there it is! Did you know that just like this cookbook, DNA also contains all of the right amount of recipe needed for the production of protein? Well, if you don't, then fasten your stomach as I serve you our best-selling cuisine, the taste of protein synthesis. Protein synthesis can be carried out in two major steps, namely transcription and translation. In the process of transcription, DNA is replicated through the help of an enzyme called RNA polymerase. It is also the process of transferring genetic instructions from the DNA to the mRNA. Transcription takes first the process of initiation. In here, the RNA polymerase binds to a region of a gene called promoter. Once it binds to the region, it signals the DNA to be split into two so that the enzyme can read the bases in one of the DNA strands. Once it's split, the enzyme is ready to make a strand of mRNA with a complementary sequence of the bases. The process is then followed by elongation. This is where the addition of nucleotides is placed into the mRNA strand. Eventually, the process ends with termination. In this step, the mRNA strand is complete and is ready to be detached from the DNA to further go to the process of translation.
Hold up right there! It's still not over yet! To make sure that everything is correctly done, the mRNA strand that has been transcribed will still need to go through further processing before it leaves the nucleus.
This processing includes splicing, where it removes introns from the mRNA where it do not code for proteins. Next is the editing, where it alters the nucleotides attached to the mRNA. Finally, the polyadenylation, where it adds a tail to the mRNA that signals the end of the mRNA.
In translation, mRNA along with ribosome and transfer RNA works together to produce proteins. The process starts with small ribosomal subunit attaching to the mRNA molecule, and at the same time, the initiation tRNA recognizes the mRNA sequence and binds to a specific codon sequence on the mRNA molecule. The large ribosomal subunit will then join the complex. The tRNA resides in one of the binding sites in the ribosome called P site, leaving the second binding site, the A site, open. When a new tRNA molecule recognizes the new codon sequence in the mRNA, it will also attach to it and occupy the A site. A peptide chain will then form connecting the amino acid of the tRNA in the P site and the tRNA in the A site. As the process moves, the tRNA from the P site will then be released, translocating the tRNA in the A site to the P site, leaving the A site empty again. Another molecule of tRNA will soon occupy the A site. The process goes on until the amino acid chain reaches a termination codon on the mRNA. When this happens, polypeptide chain is made and released from ribosome. The polypeptide chain is also known as the protein.
Why is this important?
Protein, just like this albumin from the egg white, is the building block of life. Every cell in the human body contains protein. Without it, just like a falling building, everything will collapse and will leave no existence. Sugar, spice, and everything nice. Now that your stomach is satisfied, would you dare go taste for another slice?